Hi, my name is Dagmar Timmer. I'm from Essen in Germany, and together with my colleague Andreas Deistung, I would like to present data of a study we have recently published in the journal Brain Communication. Quantitative susceptibility mapping, QSM, is a useful tool to study brain iron concentration in vivo. Iron accumulation is known to play a role in degenerative diseases like Huntington's disease, Parkinson's disease, or Alzheimer's disease. Although the iron content is known to be high in the dentate nuclei, the most important output structure of the cerebellum, surprisingly little is known about possible changes in iron content in cerebellar ataxias. The only exception is Friedreich's ataxia, where a lone frataxin level is known to be associated with changes in iron metabolism. The aim of our study was to use QSM in order to visualize the deep cerebellar nuclei in different forms of the cerebellar ataxias and to quantify volume and possible changes in iron concentration. We studied patients with spinocerebellar ataxia type 1, SCA or SCA1, SCA2, SCA3, and SCA6. These disorders are the most common forms of dominantly inherited ataxias. We also studied patients with Friedreich's ataxia, the most common hereditary ataxia, which is a recessive disorder. And we included one form of sporadic cerebellar ataxia, multiple system atrophy from the cerebellar type.
My name is Andreas Deistung, and I am from the University Hospital Halle. Within the study, we performed MRI examinations at a magnetic field strength of 3 Tesla and clinical examinations for gathering clinical history, genetic diagnosis, as well as neurological scores. Based on quantitative susceptibility maps derived from the MRI examinations, we specified the dentate nuclei to study their volume and susceptibility on a volume of interest based level. Further, we carried out voxel-based analysis of the susceptibility distribution and analyzed the volumes of interest of the dentate nuclei using a voxel-based morphometry approach. Here we see an axial section of the dentate nuclei showing the results of the voxel-based group analysis between individual ataxia subgroups and their corresponding controls. The first 2 rows show the average susceptibility maps for the patients and the corresponding control groups. The absolute difference between mean susceptibility maps between patients and controls, as well as voxel-wise statistical comparisons, are shown in the 3rd and 4th row respectively, indicating significantly higher susceptibility values in SCA1 and multiple system atrophy of cerebellar type patients, moderately higher susceptibility in Friedreich's ataxia, and significantly lower susceptibility in SCA6 patients when comparing to the corresponding control groups. The findings of the voxel-based analysis are supported by analyzing the susceptibility values within the bulk dentate area. Here we see the statistical analysis result of voxel-based morphometry of the silhouette and bulk of the dentate nuclei in the 3rd and 4th row respectively. The most striking volume reductions were seen in SCA6 patients. In SCA1, SCA2, Friedreich's ataxia, and multiple system atrophy of cerebellar type patients, there were significant reductions in the silhouette and bulk of the dentate, but much smaller compared to SCA6 patients. Almost all of these findings on dentate atrophy are supported by volume of interest-based analysis. We could show that quantitative susceptibility mapping revealed abnormalities of the dentate nuclei in common types of cerebellar ataxias. higher susceptibility in the dentate nuclei observed in SCA1 and multiple system atrophy of cerebellar type patients suggest a reduction in neurons being associated with an increase in iron concentration and or an increase in iron-rich glial cells, for example due to microgliosis. Demyelinization may also contribute. The lower susceptibility measured in SCA6 patients confirms previous studies and suggests a loss of iron-rich glial cells. Magnetic susceptibility data warrant future studies of iron content and iron-rich cells in the dentate nuclei to gain a more comprehensive understanding of the pathogenesis of cerebellar ataxias, as well as to explore additional biomarkers and treatment options. For more information and details, please check out our publication at Brain Communications. Thanks a lot for your interest.